So the title of today's talk will, sorry, where's my screen?
We're actually seeing your screen.
Can you see my screen?
Yes.
Okay,
Yeah.
I didn't see my screen very interestingly. Oh, okay, now I can see it. Okay, so the title is Nutrient-Driven Histone Code Determines Exhausted CD8 T Cell Fates. So we always talk about you are what you eat. At a cellular level, a cell's identity and function are usually determined by the nutrient metabolism within the cell, which provide the donors or cofactors for the epigenome and consequently regulate their epigenetic states and cellular differentiation. However, what we really don't know is whether this nutrient metabolism can directly influence the epigenome and consequently regulate the cell phase. In that words, we want to ask, do a nutrient-driven epigenetic code actually existed in cell phase? And today, the story I'm going to tell you is really about it is a nutrient-driven epigenetic code that has been involved in CD8 T cell differentiation. And as CD8 T cell differentiates, the types of the nutrients they metabolize actually can directly involved in regulating their epigenetic states and consequently regulate their cellular function. In particularly, I'm interested in the antiviral immune responses and anti-tumor immune responses. So now I would like to give you a brief introduction about T cell differentiation. So during chronic infection and in cancer, naive T cells will gradually differentiate into these early effector cells. And these cells will further differentiate into these progenitor-like exhausted T cells. And these are the type of the cells, they still maintain certain proliferative capacity and cytokine producing ability. And I would also want to remind you that these are the types of the cells that they can respond to immune checkpoint blockade. And these are the type of the cells we really want to enrich during our current immune therapy. And as antigen persists, these cells will gradually differentiate and gradually lose their proliferative capacity and cytokine producing ability. And eventually they will become functionally exhausted, which we call them these terminally exhausted T cells. And very interestingly, these two types of the cells has been shown that they also have very distinct nutrient metabolism, as you can see here. And furthermore, when we perform ChIP-seq analysis for A3K27 acetylation, which usually indicates the activation of the genes on these two types of the cells, you can see that these two types of the cells also have very unique histone acetylation landscape, as you can see here. And the histone acetylation usually use acetyl-CoA as their donor for this modification, which directly derives from the nutrient metabolism within the cells. So this link actually leads us to ask the question, how do this acetyl-CoA and its upstream nutrient metabolism could contribute to this distinct epigenetic states that we saw in these two types of the cells and consequently regulate their cell states and differentiation. And in mammalian cells, acetyl-CoA can be produced from different types of the nutrients, such as glucose and acetate, through different metabolic enzymes, including ACLY and ACSS2. And these acetyl-CoA will be further translocated to the epigenome and consequently regulate their gene expression and function. However, what we really don't know in the field is whether if it is this total pool of acetyl-CoA that matters in driving these epigenetic changes, or the source of acetyl-CoA matters. So if the source matters, we want to ask the following question. First, as T cell differentiate, do they have their own preferences in utilizing these different types of the nutrients? And secondly, whether these acetyl-CoA that derived from this different type of the nutrients can be allocated to the distinct locus of the genome and consequently regulate the gene in a locus-specific manner. And we would like to call this a metabolic code. So if this cell really existed, these types of metabolic codes, can we learn this code and try to reprogram CD8 T cells through a nutrient-driven interventions? So to ask all of these questions, we have been first focused on these two key metabolic enzymes, ACLY and ACSS2. We profiled their expression pattern of ACSS2 and ACLY in mouse CD8 T cells from tumors. And very interestingly, I hope you can see here that as these T cells gradually lose their functionality and become exhausted, there is a significant drop of ACSS2 mRNA expression pattern. However, the expression of ACLY stays relatively stable across the states, or even slightly higher in the most terminally exhausted CD8 T cells as compared with their functional counterparts. And this is not only true at the mRNA level, because we see a similar pattern of the significant drop of ACSS2 at the protein level as well, as you can see here. And the distinct expression pattern that we observed for ACSS2 and ACLY in this functional progenitor and dysfunctional exhausted T cells has been also correlated with their distinct utilization of either acetate or glucose for their acetyl-CoA production through an C13 tracing analysis we performed. So this very interesting phenomena in terms of this unique expression pattern of these two enzymes and their relatively preferential utilization of glucose and acetate leads us to ask, do ACLY and ACSS2 actually involves in regulating CD8 T cell differentiation and function? And the answer is yes. Because as you can see here, when we knock out ACLY, there is a significant increase for this functional progenitor formation. However, knocking out ACSS2, the cells are more trended to form this dysfunctionally terminal differentiation. And this distinct regulation of ACLY and ACSS2 in either this functional progenitor or this dysfunctional exhausted T cell differentiation has been further transformed into their unique role in anti-tumor immune responses, as you can see here. So here, when we knock out either ACLY or ACSS2 in CD8 T cells, and then transfer them into a B16 GP33 tumor-bearing mouse. And here, the GP33 is an antigen that could be recognized by the P14 T cells. And after day 21, you can see very strikingly knocking out ACLY in CD8 T cells can significantly inhibit this tumor growth as compared to the control. However, knocking out ACSS2 shows totally the opposite effect, that the tumor grows a lot bigger than the control. And this is not only true in mouse, since we see a similar phenotype in human CAR T cells. So in the bottom line, you can see here, a similar setting has been applied when we knock out ACLY or ACSS2, but in human CAR T cells, and then transfer them into an NSG mouse. And consistent with our observation in mouse, you can see that knocking out ACLY in these CAR T cells, the tumor become a lot smaller. However, knocking out ACSS2, the tumor grows a lot bigger as control. So now the question comes, since we already mentioned to you, ACLY and ACSS2 are both important for acetyl-CoA production. But if you think about that, why these two key enzymes that have the similar function sounds like, but why they have such a unique role in regulating the differentiation and the function of CD8 T cells? Is that possible because this acetyl-CoA that derived either from ACLY or ACSS2 can be distinctively allocated to the distinct genome locus of the genome and consequently regulate the gene in a locus-specific manner? And to address this question, we performed the cut and tag analysis in wild type and ACLY knockout T cells for A3K27 acetylation, A3K9 acetylation, H4 acetylation. So as you can see here, very consistently, these cells form two very distinct unique clusters across the three modifications. And in particularly, the peaks that has been enriched in ACLY knockout T cells has been mostly correlated with this functional progenitor differentiation. However, the peaks that lost in ACLY knockout cells are mostly corresponding to this dysfunctional terminal cell differentiation. So when we further perform
One moment.
motif analysis on these peaks, we see that the peaks that enriched in ACLY knockout T cells are mostly regulated by this AP1 BZIP transcriptional factor family member.
Tijin, I think we are, you have a, you have to disconnect your AirPods.
Members. So from this data, it really shows that ACLY do regulate this T cell differentiation through an epigenetic mechanism. And then it comes to a next question. Since ACLY and ACSS2, they are both metabolic enzymes, how can they regulate these epigenetic states in a locus-specific manner? Is that possible that these enzymes, ACLY or ACSS2, could specifically interact with histone acetyltransferases, which consequently regulate the locus-specific gene expression in the individual cell subsets? And secondly, can we try to learn this kind of protein coding complexes and reprogram these most terminally exhausted T cells into their functional states? So to answer all of these questions, we have first screened all the hats within the CD8 T cells. So very interestingly, as you can see here, we saw that knocking out P300 phenocopies ACSS2 knockout in enhancing this dysfunctional terminal cell differentiation. However, knocking out another histone acetyltransferase, CAT2A, mimics the observation from ACLY knockout in promoting this progenitor cell differentiation. So this data do suggest us probably there are two protein complexes in either this functional progenitor by ACSS2 P300 or this dysfunctional exhausted T cells by ACLY CAT2A to regulate their differentiation. So our next question is whether this potential functional correlated protein complexes can physically interacting together and thus regulating their cell fate differentiation. So to answer this question, we performed a proximity ligation assay in either these functional progenitor cells or these dysfunctional exhausted T cells for the interaction of ACSS2 and P300 or ACLY and CAT2A. And as you can see here, very interestingly, ACSS2 and P300 preferentially interact with each other in these functional progenitor cells. However, as these cells lose their functionality, ACLY predominantly interact with CAT2A for their interaction. And thus now, combine the data that I showed you and also earlier, we do think we found these very unique two protein complexes within either this functional progenitor and dysfunctional terminal cells, where ACSS2 and P300 can interact together to regulate most like a functional effector gene expression in these progenitor cells. However, as T cells lose their functionality, ACLY and CAT2A preferentially interact for this terminal cell differentiation. And since now we already learned these two protein complexes in these two types of the cells, we now want to ask, can we try to use this metabolic code that we already learned to reprogram these most dysfunctional exhausted T cells to their functional state through an enhanced nuclear ACSS2 expression? And the answer is yes, it's possible. So what we do now is actually we try to overexpress ACSS2, the nuclear version of ACSS2 in the T cells. And as you can see, they quite localized within the nuclei and co-localized nicely with P300. And very significantly, we also show that this nuclear localized ACSS2 also significantly enhanced the histone acetylation landscape for the most functional progenitor gene expression as compared for their dysfunctional counterparts, wild-type counterparts. And consequently, these ACSS2 overexpressed cells has been significantly inhibit this tumor growth as compared with wild type, indicating that by overexpressing this nuclear ACSS2, we could potentially enhance their anti-tumor immunity. And now, coming back to our original question we have been asked, you are what you eat. So I hope by what I've been showed you today, you are convinced that at least in CD8 T cells, their differentiation and functionality are also determined by the nutrient metabolism they utilize. In particularly, when they are in a more functional state, they preferentially utilize acetate through ACSS2 P300 protein complexes to regulate their effector-related gene expression. However, when these cells lose their functionality and become exhausted, they preferentially utilize glucose-derived citrate through ACLY CAT2A complexes for the exhaustion-related gene expression, and consequently regulate their cellular states. And also, by learning these two protein complexes, we showed that potentially in the future, we could reverse these dysfunctional states by an enhanced nuclear acetyl-CoA production. So while this study that I have been showed you today has been majorly focused on CD8 T cell differentiation, I hope you can also appreciate that it brings us a broader paradigm in understanding how these nutrient preferences in cells can regulate this local specific gene expression and their cellular differentiation. And with that, I would like to thank my mentor, Sue, and also my collaborators, Rusty Jones, Shaosun Zhang, who has been helping with all this metabolism-related work, and also Yingxiao Wang, who has shared a lot the construct for the CAR T. And then